knowledge and skill of the dental surgeon and their role in management, acute and chronic pain, oral mucositis in the oral cancer, and regeneration in oral and dental tissues. It gives me immense pleasure to be among the most esteemed personalities who have won accolades in their respective fields. So let's begin with the blessings of Almighty, the very first webinar of this series, of the international webinar series of SANKALCHAND PATEL University. I owe in awe with the collected expertise of the gathered here, those all who are gathered here. We are deeply honored to welcome all the respected, esteemed, and knowledgeable dignitaries who are present with us in this online series. Now, to deliver the welcome speech and a brief introduction, I would like to request Dr. J.R. Patel, sir, Dean, Narsibhai Patel Dental College and Hospital, SANKALCHAND Patel University. Sir, please.
Good evening to all delegates from India, and good morning to our eminent speaker, Dr. Praveen. For this webinar series, today's mentor, Dr. Praveen R Arany, who is from Oral Biology and Biomedical Engineering, School of Dental Medicine, Engineering and Applied Sciences, University of Buffalo. Dr. Anil Managutti, who is heading the Department of Oral Surgery, and his team members, Dr. Shailesh Mehnat, Dr. Rushit, Dr. Nirav, Dr. Disha, all other faculty of Narsibhai Patel Dental College and Hospital, and all dear delegates. With proud privilege and pleasure on behalf of SANKALCHAND Patel University and myself, I welcome you all for today's International Multidisciplinary Webinar on Introduction to Photobiomodulation Therapy, a powerful tool for regenerative clinical dentistry, which is organized by Department of Oral and Maxillosurgery, Narsibhai Patel Dental College and Hospital, which is a constituent institute of SANKALCHAND Patel University. The whole program is organized under the leadership of Dr. Anil Managutti by the Department of Oral and Maxillofacial Surgery. There is a golden opportunity to learn about new concept of photobiomodulation therapy from our eminent international speaker, Dr. Praveen. I congratulate the whole organizing team for such an important webinar series, the first session of which today we are going to have. I hope there will be a definite take-home points for all attendees. Thank you. Wish you happy webinaring. Jai Hind.
Thanks a lot, sir. Thank you. And now it's my great pleasure to invite Dr. Praveen R Arany. Dr. Arany is trained as a dentist and an oral pathologist. He completed a joint Ph.D. residency program at Harvard University. He has 2 certificates in clinical translational research from Harvard Medical School and National Institute of Health. He pursued postdoctorate fellowship at Indian Institute of Sciences, Bangalore, National Cancer Institute, Bethesda, the Harvard School of Engineering and Applied Sciences, and VISNAGAR Institute, Cambridge. He has over 80 scientific publications and is a recipient of numerous awards. He pursued postdoctorate as well as he has also been invited to speak in various national and international forums. Besides his academic interest in clinical translational research, Dr. Arany has co-founded 2 companies on the dental and biophotonics product to actively translate his lab research into the real-world solution as product and technologies. So I humbly welcome Dr. Arany to please begin his speech.
Can you hear me? Yeah. Right, let me share my screen before I start. Can you see my slides?
Yes.
Great. Well, thank you so much for the invitation, Dr. Anil Managutti, a good friend and a colleague from my dental days. Dr. J.R. Patel, the Dean, for your kind invitation, and Dr. Ankita for that kind introduction of my background. So I am thrilled and honored to be the first speaker in this webinar series, and I hope the Institute makes many more of these, because I think the bridge between what's happening in research needs to make its way to clinic, especially a lot of the research that we do in clinical translational research. Via these webinars, there's a really good way of transforming it into clinical care. So thank you for the invitation, and I hope I can provide some information about this new treatment, photobiomodulation. So the title of my talk is an introduction. I'm assuming that many of you have heard about different types of light treatments and lasers in dentistry in the bigger scope. But I'm going to introduce you to one particular kind of treatment, which is a non-surgical, non-thermal use of light to stimulate wound healing and tissue regeneration. These are my disclosures. My research is supported by various companies and federal and state research funds. There is also support from various organizations. There are, as you can see on this board, many international and national organizations dedicated to the use of low power laser and light to do treatments. And it is also called low level light treatment, and it's also called photobiomodulation. As you can see from this board, there are various organizations whose prime reason or purpose is to try and bring this science to clinical care. So I will begin my talk by, I think, trying to connect the dots for us. All of us are trained as dentists. Well, most of us in the audience are now clinical dentists. And dentists are very good at replacing tooth structure, right? We are very good at replacing tooth, hard tissue. We are very good at replacing soft tissues. And if you look at some of these images here, they are such beautifully done restoration and prostheses that in any other field, we would put them in a museum, right? They're such beautifully done, very nicely, elegantly replacing normal tooth structure. But that is a limitation, because we are getting better and better at replacing tooth structure, but we know that it is prone to biomaterial failures over time. So the process of wound healing, where you're able to restore a normal biological tissue, and all of us have read about this in our basic physiology and pathology, where we know that wound healing occurs in different phases. When you get a tissue damage in any area, including your mouth, you are subjected to various clotting factors, hemostasis, followed by inflammation, proliferation, and reorganization. And we have studied this in much detail. So there is a very good biological understanding of what occurs during tissue healing. We also know that the ultimate outcome of good healing is tissue regeneration. And the difference between tissue regeneration and healing is basically the ideal completion of the healing process, where you can restore both form and function. And we have learned a lot about regeneration from the two basic processes. You probably heard about the amphibia, or lizards, if you will, that can regrow their limbs. And then there is this flatworm or planaria, where we know that when we cut them into smaller and smaller pieces, the whole organism can develop from a smaller piece. So these are two different processes, epimorphosis and morphollaxis, and there are dedicated folks who are studying what causes this regenerative process. So this is something that we all strive to do in our regenerative clinical practice, but how do we go about doing it, right? So when I was a student in pathology with Dr. Anil back in KLE Belgaum, we were studying different processes. We were learning about wound healing, we are learning about embryonic development, and then we are learning about cancers, right? Different types of cancers. And I just told you about the different phases of wound healing. Interestingly, when you look at wound healing, the early parts of the wound healing are very similar to cancers, because they have a lot of proliferation, migration, and differentiation, or de-differentiation in many cases. So the big difference between cancers and development, which occurs during the embryonic formation, is the control of the biological process. One of the major disruptions to this is seen when you're undergoing wound healing. So if you look at the very early parts of wound healing under the microscope, or even histologically or clinically, the early wounds are very disorganized, right? And it is very tumor-like. But over time, there is a setting in of a biological regulatory process which converts the healing response into normal tissue, almost embryonic development like. So this was the primary motivation for my research, which I started about 2 decades ago in India. And I have continued to think about how it can be controlled wound healing in the hope that someday we can control tumors, right? So this is the rest of the story. And the reason that we want to understand mechanisms of why this thing works is so that we can control clinical outcomes. If you know how a thing works, then you can use that information or knowledge to control clinical outcomes. So the motivation for the rest of my talk that I'm going to tell you today is about how can we control wound healing and what are the tools at our disposal. So all of you are very familiar with different kinds of biological regulation, right? You have, I'm assuming there are some periodontists in the audience, there are orthodontists and oral surgeons. I know Dr. Anil is an oral surgeon. So we are all very familiar with growth factors and grafts and other kinds of small molecule drugs that can promote wound healing. Today, I'd like to introduce you to the concept of light. And the basic premise of today's talk is, can we use light as a drug or as a medical treatment? So hopefully by the end of this talk, you will have some information to put this statement in context, where we will try to explore the use of light as a treatment. If you think about light as a physical entity, it is the most fascinating physical constant in the universe, right? Everything from light years, when you're trying to measure the distance between stars, to the very basic force between two subatomic particles. If you've heard of the string theory, there is a hypothesis that there is a nature of vibrations that simulates light-like forces. So we know that light can impact the smallest of scales, and we know that light is important for the largest of scales. Interestingly, today, we will talk about how we can use light in an intermediate scale, everything at the level of cells, tissues, and hopefully the complete organism, the human body, right? So how can we use light in these processes? When I give these talks, I'm always asked, we are not plants, right? Human beings are not plants. So how can we use light in human health? And sure enough, there are various examples that we have studied in physiology about how sunlight can affect vitamin D metabolism. We know the precise biochemical steps that are necessary for the conversion of pre-vitamin D to vitamin D, which maintains our bone and tooth health, right? We know that very well. We also know that your very ability to see these slides is converting visible light energy into a biological signal in your brain that is perceived as vision. And we know the precise biochemical steps that are necessary in the retinal pigmental epithelial cells, in your cells of the retina, that is containing this molecule rhodopsin, which then senses light and changes its conformation to induce biological signaling. All of us are very familiar with the use of light and how it affects our circadian rhythm and our psychological state. So we know that dark places are not good for our overall mood, while we are also very familiar when we travel with jet in various, overnight travel over with the jet plane, we actually get jet lagged. And that is, again, a disruption of our circadian cycle. So all of these are great examples of how light can have an important effect on your normal health. It was this gentleman, Niels Rydberg Finsen, in the early 1900s, who actually developed a device. And this is the device that he was using to treat people. And this is basically a lens apparatus that can purify sunlight. And by using this device, he was able to partition sunlight into blue and red light using different filters and lenses. And he was able to actually cure tuberculosis of the skin, lupus vulgaris. He was actually recognized as a pioneer in the field of photomedicine and was awarded one of the first Nobel Prizes in medicine and physiology back in 1903. So this is not new. It's been around for a long time, and it was actually recognized in the early 19th century as something that is capable of helping human treatments. The concept of a laser, I'm sure all of you are aware, was proposed by Albert Einstein, and the laser stands for light amplification by stimulated emission of radiation. It took them 60 years to actually build a laser unit, and this was in the early 60s by Charles Stone and Theodore Maiman. When you invent a new medical device, one of the first things that we all do as biomedical researchers is to do a dose escalation study. We take a bunch of animals. After appropriate ethical concerns and approvals, we do experiments where we increase the amount of dose and see if there is a response. So Dr. Andrea Mester in the late 60s found that when he was using high power lasers, he was able to very nicely cut and cauterize tissue, which all of you who use lasers are very familiar with. But at very low doses, he found that he was getting stimulation of hair growth and promotion of wound healing. So this is an image of some of Dr. Mester's experiments where he was creating, if you can see my pointer here, he was creating these wounds on the back of the mouse. And when he treated them with low amounts of laser light, or specifically a ruby laser, he found that he could stimulate the wound healing in a controlled manner. So he called this process photostimulation. And over the years, there have been over 300 different terms to describe this treatment. Some of the most popular terms are low-level light treatment or low-level laser treatment and photostimulation. So back in 2014, now 6 years ago, a group of people who are well-known in the field all got together in Arlington, Virginia, and we came together to discuss what are we going to call this treatment. Because there are so many names and it's causing so much confusion, right? We did not like the term low-level light or laser treatment because we could not explain to our audience what low means, low in terms of what, and level of what, right? So we agreed on the term photobiomodulation because it represents the use of light to modulate biological processes. And the reason we like this term, and it has become standard now, is because it describes the use of a non-ionizing source of light or photonic energy in a non-thermal manner. I'll try to emphasize that again. It is a use of light in a non-thermal manner. So the reason, and this is the longer definition, which you don't have to worry about, but it talks a lot about what are the other sources of light. It does not have to be a laser. It can be an LED or a broadband light at the right wavelength and the right dose. It is a non-thermal process. And if you use this light treatment appropriately, you can promote certain positive processes, such as wound healing and tissue regeneration. At the same time, if you use a different dose in a different manner, you can inhibit certain negative processes, such as pain, inflammation, and an aberrant immune response. So the use of photobiomodulation, the term photobiomodulation, is now the most accepted term. And all of you who use PubMed, you know that it's the largest scientific database in the world for scientific literature. It is maintained by the National Library of Medicine at the National Institutes of Health, Bethesda. And when you search for a term about a disease or a treatment, we use something called the medical subject head term, a MESH term. So photobiomodulation is now the most accepted MESH term to describe this treatment. And if you want to find literature on this area, you should use the term photobiomodulation or PBM. This slide overviews many of the various treatments that are possible with photobiomodulation, and it is both a bane and a boon for our field. There are very few treatments in the field of medicine that can do everything from curing trauma to the head, concussions or stroke, to wounds on your feet and ulcers in your mouth, right? How many treatments do you know of that can do all of these different things? But all of these things that are listed here, all these applications that are listed here, are evidences from human clinical studies. And we all know that if something works in many different areas, they must have different mechanisms. So the next part of my talk, very briefly, will go over the mechanisms. And feel free to stop me at any point or at the end, and I'm happy to answer the question. I'm also very happy to provide the entire slide deck. I'll put my email at the end. If anybody's interested, I'm happy to email them this presentation. So for those of you who use lasers in clinical practice, we all use either photo disruption or high energy transfer of energy into the tissues to cause disruption of tissues. And that is your surgical lasers, either soft tissue, and now more and more we are using hard tissue lasers. You can also use light to disinfect, lasers to disinfect biofilms, and that is it is the photothermal effect of using light to disrupt microbes. There are two other treatments, and photobiomodulation is one of them, where we are trying to use photochemical reactions and not the thermal part. And there is another treatment called photodynamic therapy, which uses a combination of a dye and light to disrupt biofilms. So we are going to talk primarily about photobiomodulation. And this is a rather busy slide, but the only point I want to make here is we have understood many of the different mechanisms of how photobiomodulation works. And to summarize that, we think of them in three separate silos, or three different categories of mechanisms. So the first mechanism involves it's an enzyme present inside the mitochondria, and we all know that cells have the mitochondria, an organelle capable of mediating the metabolic energy transfer inside the cell. So there is a very specific enzyme called cytochrome C oxidase, which is present on the mitochondrial membrane, which is capable of absorbing light. It absorbs light at a very specific wavelength. And when you get light absorption by this molecule, it increases the amount of ATP, adenosine triphosphate, and reactive oxygen species, ROS. The increase in ATP and ROS in a limited amount within this particular complex results in therapeutic benefits. The second mechanism involves light-sensitive receptors on the cell membrane, especially the neurons, right? You know that transporters on the neuron are responsible for neural conduction. Absorption of light by these molecules results in disruption of pain conduction or stimuli, neural transmission, and this results in pain relief or analgesia. The third mechanism was discovered in my lab, where we found that activation of an extracellular growth factor, TGF-beta, results in activation of the biological signaling responsible for healing and tissue regeneration. So in the field, we broadly think about when you do light treatments on patients, you are treating the entire tissue. There are epithelial cells, there are fibroblasts, there are macrophages, neurons, you name it. So all the cells are being exposed to light. So we know that light will affect different mechanisms in different cells. But broadly, when you're trying to improve muscle performance, you are primarily focusing on the cytochrome C oxidase. While if you're trying to relieve pain, we are thinking about the pain transmission receptors and transporters on the neuron surface. While you're trying to promote wound healing, we are thinking about extracellular TGF-beta, the growth factor activation. So this is a really busy slide, but it tells you that because there are multiple mechanisms that you can use with light, you can do all of those different clinical treatments. But you must understand which mechanism you want to use in which context. And that is the caveat that unfortunately is still not very popular. So if there is one thing, if you have done low-level light treatment or photobiomodulation before, and you have gotten somewhat good results, but not consistent results, it is because we have to use the right dose for the right mechanism, right? And like everything like we have done, everything from amalgam to composites to root planing and scaling, unless you understand the proper technique and how to use it in the right context, you can't do scaling for caries, right? You will never do that. Neither will you do a filling for periodontal disease, right? So similarly, when you think about when you want to use photobiomodulation, you should understand what clinical context and which mechanism you want to try. So the third part of my talk will focus on one specific application. We do research in various areas, but as was mentioned by Dr. Ankita, and this was the primary reason Dr. Anil Managutti invited me, is there is now new guidelines for oral mucositis. For those of you, I assume most of you are practitioners or students in the audience. So why do we practice a particular clinical procedure, right? We have been asked to do a particular procedure. Why do we do that? It is because the evidence for the use of this particular procedure or material has been validated in basic science studies, but also in clinical studies. So you have all seen this pyramid of evidence where the lowest level evidence is an opinion. Any one of us can have an opinion about how to do things. But if you can see case reports and case series from various clinical studies, we know that it works in patients. Once we have that level of evidence, we organize clinical trials, and we do that at my center, and my lab specifically leads PBM clinical trials. We looked at organizing multiple centers that will do photobiomodulation clinical studies, and then we sit down and collect the evidences from all of them and do a systematic review and meta-analysis, right? You have heard these terms before. So for those of you who are very familiar with evidence-based dentistry or medicine, we follow this pyramid of evidence. And only when we reach the highest level of evidence, which is a systematic review and meta-analysis, only then do we write clinical practice guidelines. You have to do a lot of work both in the lab and the clinic before you can actually change clinical practice. And that is where photobiomodulation currently is. In some diseases, not only do we have very good basic science studies, but we also have great clinical studies that have reached the level of evidence where we are changing clinical practice. So all of you are very familiar with mucositis. We know that patients who get chemo or radiation or even transplants present to our dental clinic with ulcers in their mouth, right? It can be as simple as redness or erythema to outright breakdown of the tissue and ulceration and wounds. And this is just the WHO scale. Unfortunately, while we are trying to treat the cancer in a patient, this is a significant reason for loss of quality of life and morbidity in patients. Sometimes, especially grade 3 and grade 4, the mucositis is so severe that the patient will refuse to do further cancer treatment. So not only is it a quality of life problem, especially for a cancer oncologist or a surgeon who's trying to do treatments, it can also be pretty lethal. And that is why this treatment, unfortunately, this disease, unfortunately, has to be dealt with by dentists and as a part of the oncology treatment. So if you look at these slides, you know that it is red, you know that it is inflamed, and you know that there is a wound. So all the things that are the basis for this disease, pain, inflammation, and lack of healing, like I told you just now, photobiomodulation has been shown to be effective. Right now, if you're seeing these patients, we use either bland mouth rinses or combination of either an analgesic or an opioid. And people have actually mixed this together. And I know there are various recipes for these, but a magic mouthwash is basically a combination of all these agents. There are also certain molecular therapeutics, such as keratinocyte growth factor or fibroblast growth factor or epidermal growth factor, that are also used in severe cases. And even in the US, not just in India, I think these are very expensive and they are very effective, but they are pretty expensive. And they have been limited success because of that. So back in 2013, a group of experts in oral medicine, oral biology, radiation biology, and oncology came together and reviewed the literature for the use of photobiomodulation or low-level light treatment in the management of oral cancers, oral mucositis. In 6 years after that, very recently in the late 2019, I was a part of that group that actually re-reviewed that literature. So back in 2013, the evidence was not as strong as it is today. It was suggested as an adjunctive treatment. But now the evidence is strong enough over all these 35 different studies that we have reviewed that this treatment should be used as standard of care. So this is a systematic review and meta-analysis. And we were able to show that PBM should be used in both prevention and treatment of oral mucositis. So this is something that you will get a lot of your oncology colleagues, especially head and neck pathologists, head and neck radiologists, who use radiation to treat oral head and neck cancers, they will come to you and say, can you do photobiomodulation for me as a dentist? And this is the literature on, or this is the source literature on which they are recommending the use of photobiomodulation. So why does photobiomodulation work in this particular case? And I'll show you 2 slides. And again, I'm happy to share the entire presentation if you're interested. We talked about different mechanisms on why photobiomodulation works. And this is basically the references for the use of photobiomodulation, which has both primary and secondary effects. So the initial effect of doing the right dose of light treatment on these tissues is that it causes a depolarization of the nerve conduction. It is causing pain relief directly by modulating certain transporters. If you're familiar with this term, TRPV1, they are responsible for reducing axonal transport of mitochondria within the neurons. So all of these are initial effects when you do light treatments, and those are the references on your right side where this evidence has been shown. If you wait a little longer and you look at the changes in the patient's physiology, we have also been able to measure changes in circulating markers of pain perception, such as serotonins, beta endorphins, as well as all of these other markers. So when you do light treatment for pain relief, you have an initial reaction in the tissue, which prevents nerve conduction. And over long term, it also is able to elevate levels of pain fighting substances in your body. Both of these are responsible for the analgesic effect. One of the concerns of using light for analgesia is that people ask us all the time that if you're using a laser, will you cause any significant damage? And the answer is an emphatic no. The amount of light that we use for photobiomodulation in analgesia is a non-thermal dose, and it is completely reversible and transient in nature. It is very similar to using your local anesthetic, lidocaine or bupivacaine or any of the other local anesthetics. So none of them have persistent analgesic effects, right? You don't get paresthesia forever if you use local anesthesia. And similarly, if you use the right dose of photobiomodulation treatments, you should not get any persistent paresthesia. It's completely reversible and it is transient in nature. The other reason, as we saw in that initial slide with mucositis, is that you get an increase in the healing response. And again, I will not belabor the point. We have outlined several mechanisms that are responsible for the stimulation of the healing. I talked about TGF-beta. That's a growth factor that is present outside the cell. This growth factor, when it's activated by light, is capable of healing the tissues. We also, I'm sure some of you have heard of the term NF-kappa B, which is, again, a central player. It's a molecular signaling pathway that is central to both pain and inflammation. And we have shown, our group and other groups have shown, that when you use light in a controlled manner, you can activate this factor and then do its analgesic and anti-inflammatory effects. The clinical evidence is great, and all of us are, most of us are clinicians here, so we like the clinical evidence, but having access to a basic science lab, we like to ask basic science questions. So we like to see the evidence in our own hands, right? So let me show you very briefly 2 slides that talk about the kind of experiments we do. So we are an oral biology lab and we have access to oral tissues. So we've taken oral keratinocytes, which are oral epithelial cells. And if you look at that top panel there, we put these cells in a monolayer. We are seeding the cells completely in a plastic dish, and then we make a scratch. The scratch represents the mucositis wound that we create. And as you can see in this panel, in the second and the third panel, over time, the cells will migrate and close the wound. This is a normal healing reaction. If you use photobiomodulation, and this is different doses, 0.1 watts and 0.5 watts, you can see that this wound is closing much, much more quickly, right? You can see that at 24 hours, it's smaller than the ones that normally heal. And at 48 hours, it's completely closed, while it's still a little more persistent in the normal case. This basically demonstrates that the use of light is capable of accelerating the epithelial closure and aiding healing, which is what we expect in our patients. It is also very important to point out that if you do not use the right dose, as you see in the lowest panel, you will not get that therapeutic response. In fact, you can see that the healing is slightly delayed. So like every tool, whether it's a scaler, whether it's a bar, whether it's a surgical tool like a laser, you must understand how to use it and the protocol to use it. And that is very, very important before you start clinical treatments. Here's another experiment where I hope you can see these. You can see these small dots of cells, right? So these are epithelial colony forming units or epithelial stem cells. So when you know that when you get a wound in the tissue, there are cells at the site and around the wound, which have stem cells in them, normally present adult stem cells. And when you stimulate them with various agents, including light, you are capable of increasing their number, which then assist in closing of the wounds. So if you use the right amount of light in the right manner, not only can you increase the acceleration of the closure of the wound, you can also increase certain stem cells in the surrounding area that are capable of promoting healing or regeneration. All of this is obviously done in vitro in cell culture. We also do animal studies with appropriate protocols. And this is a model where we are using burn wounds. So we are creating controlled burns on animals. And as you can see in the top panel here, we can see healing in the animal when the normal healing is occurring. But when you do the photobiomodulation treatment at a particular dose, this healing can be significantly accelerated. Now, we have done the same experiment with surgical wounds, which do not have the level of inflammation. You can see all that pink tissue, pink or red colored erythema around these wounds. And for those of you who have experience with burn wounds, you know that the burn wounds has a lot of inflammation, right? So photobiomodulation here not only is reducing the amount of inflammation, it is also promoting the healing response. So it is data like this from both basic science and clinical science that makes us very confident that this treatment will work very effectively in your patients. For a practicing clinician, the next question is, how do I use these treatments? And in the US, I'm sure in India as well, a lot of my colleagues in India are using soft tissue tools. Here are some examples that are available in India, and some of them are also, they're available in the US, but they're also available in India. I think this bias is very popular. All of these units are used for surgical tissue procedures, but if you use an adapter, you can defocus the beam and use it for photobiology. Given the importance of photomodulation in cancer, then you can also invest and as an InstaCare outcome. So you can use this exclusively for photobiomodulation. There are also various published protocols, and here's an example of one of the units, which tells you how to use the light and where to place the probes for how much time. So not only do we have devices that can be used right now for prevention and treatment, you also have protocols that you can learn. This treatment is getting a lot of attention all around the world, but especially in the US, which has an insurance-based medical system, we have been invited to various organizations, and this is one of the US congressional briefings that we gave, where we are showing the evidence for the use of photobiomodulation as a pain relief mechanism. So you all are very familiar with opioids and how they are wonderful drugs in the clinic, but the abuse of these drugs has caused an epidemic. So there is a lot of interest in using photobiomodulation and treatments. There is in fact now a policy, a public law policy in the US, where many government agencies are looking at non-pharmacological means of pain relief. And if you have heard about trans-electrical neural stimulation, TENS, photobiomodulation, that's the use of microcurrents. Both TENS and photobiomodulation are being used, are being explored as alternative pain treatments to relieve pain. So you're going to hear a lot more even from the policy people, not only the scientists and the clinicians. Now, one of the practical aspects, given that the audience is primarily dentists, is that we are going to be treating the head and neck area to prevent mucositis. So the other question that we very often get is, what are the side effects of this treatment? And if you have a head and neck or an oral cancer, when you're doing this treatment, will I, by any chance, increase the amount of cancer cells, right? We don't want to do that in any scenario. So although light is a part of the electromagnetic spectrum, light does not have enough energy to cause DNA damage. It doesn't have enough linear energy transfer. It is capable of thermally destroying tissue if you use a lot of energy, but it is not capable of transforming tissue. So it is not the tissue in the area around that treatment, but it's the area surrounding the initial damage. And we want to ensure that there are no sublethal damages in those regions. So the question that we ask in the lab is light treatment genotoxic or mutagenic? Can it cause mutations? And I'll show you two experiments. The first experiment here is we take these cells, in this case, they are fibroblasts that we are treating with ionizing radiation. So you know that gamma radiation is capable of causing DNA strand breaks. That's why we use it for radiation treatment. And we are measuring this molecule H2AX. And as you can see in this picture in the middle panel, there is increased DNA strand breaks when you do ionizing radiation. When you do the same experiment with lasers, either in vitro in these fibroblast cultures or in the animal skin, so this is again example of an animal study, we do not see DNA strand breaks. So you can see in the panel here that there is no green color, which indicates that there are strand breaks. So this molecule is not activated. And similarly, in the mouse skin, you can look at the area around the hair follicle, where in gamma radiation, you can see a dark brown staining, indicating that the H2AX has been activated, and this is the lung in the same animal. So you can see that when animals are treated with ionizing radiation, they get lots of DNA strand breaks. But when you use various doses of laser light, you do not see this effect. So this is the evidence that clearly shows that light treatments are not capable of causing genetic damage, gene damage, genotoxicity. We also use another assay called the Ames assay, where we are looking at transformation of a particular bacterial cell strain. And this is just, again, a very, very standardly used assay in the lab to look for mutations in different biological targets. And as you can see in your left side, this is the normal rate of transformation in these Salmonella typhimurium strains. And this is just a positive control showing that if you have a mutation agent, what do you expect to see? And as you can see from this, even when you use different amounts of light, you do not get transformation. Again, showing us that light treatments do not cause mutations or gene toxicity. This, and again, this work is published in this particular article, so you are welcome to see it. And there are various groups around the world that have shown the safety of using light for these treatments. The effect on cancer cells is very interesting. So cancer cells and normal cells at the molecular level are not very different. The major difference between them is the ability to regulate its biological processes. So we know that cancer cells have different metabolism, they have different adhesion properties, they grow in an uncontrolled manner, and they can migrate in an uncontrolled manner. So when you expose both normal cells and cancer cells to light, a form of energy, they interpret the energy in different ways. So we have found, and some of this is published and some of this is ongoing, that light treatments on cancers results in suicide or apoptosis or autophagy in cancer cells, while in the normal cells, you get proliferation and healing. So the differential response is because even though they get the same energy, they are wired differently, and therefore they receive the energy and respond differently. We also know from various publications that our natural ability to fight cancers, our anti-tumor immune surveillance mechanisms, are also stimulated by photobiomodulation. So again, all of this literature is available in this publication, showing us that both basic science and clinical evidence point to the safety of this treatment, even if the patient has cancer. Nonetheless, we do not recommend treating photobiomodulation on top of a tumor directly. So we still, if your patient has head and neck cancer or oral cancer, you can use it in a preventive manner before they get radiation or chemo, but we do not recommend directly illuminating the tumor. So for those of you who are interested, this is my email at the bottom. We have in the field started organizing our thoughts around how to use light as a drug, a photoceutical. And we are using very common concepts of drug development. So when you are trying to develop a new drug, we think about pharmacokinetics or pharmacodynamics. It basically refers to what the body can do with the drug, or light in our case, or what the light does to the drug. So these are very, very common concepts when you're trying to develop a new therapeutic. And when you think about photobiomodulation, many of these parameters have been outlined at various levels. So what is the light doing to different biological targets, as well as a clinician, what are the critical parameters that you need to pay attention? What kind of wavelength, how much dose, how many times should I treat, and how much time should be each treatment? So all of these are important parameters that you should be paying attention. So I'd like to conclude my talk by telling you that this is an extremely exciting area where dentists can actually help oncologists and your cancer treatment team. And oral surgeons also are a big part of that, and they work with various oncologists and radiation biologists. So unfortunately, the complications are dealt with by dentists in the community or in the hospital setting. So understanding photobiomodulation is really, really important. And I hope this webinar opens up many, many questions, and I'm happy to answer what I can. There is a lot of ongoing research about light-tissue interaction and how light is interpreted by biological systems. There is also large clinical trials that are ongoing, and I would encourage your team and your institute to look into trying to promote this aspect. And in the end, I'd like to acknowledge my team. These are the people who work in my lab, who actually do all the work. I get to present it. And these are my mentors from my previous labs, various collaborators who have provided various reagents, and this is my funding support. So I just end with some of the images of my lab. My email is at the bottom. So if anyone is interested in the presentation or have questions, I'm happy to take them. And thank you for a patient hearing.
Thank you, sir. Thank you for the wonderful presentation, which has opened up a great, you can say, the great knowledge aspects for us and all our delegates. Thanks a lot, sir. And now the next session is the question and answer sessions. If I feel that if any of the delegates, they want to ask few questions to such an eminent speaker, it would be a great.
It's a good chance for our delegates. Huh?
Yes, sir. It would be a very good chance for the delegate, and they can avail a lot of knowledge by asking the questions to Dr. Praveen.
I have lost my video feed, but I can hear you guys.
Yeah. Any
now
questions, participants? Yeah. Any questions by the participants?
Hopefully it was. Hopefully, sir, it was.
Hello.
No one have questions.
Dr. Anil.
Yes, sir.
Hello.
Yes, sir.
Praveen, I am Dr. Shivanand. He was my batchmate in KLE Belgaum in anatomy dissection. I want to ask one question. This regarding what is the cost of this unit to install in the college?
Yeah, that's a great question.
What cost of this unit? Photo.
So I think that's a better question for people from India, because here the units can be as affordable as I think the lowest unit we have here is $3,500 US dollars. But I think there are other units that are available from US and non-US sources. I believe the last time I visited India, there were lots of units from, I believe, Korea and Japan. So there are other units that are available, Dr. Bhagewari, but there are.
Okay.
Yeah, I don't know the exact cost in Indian rupees, but there are Indian manufacturers.
Whatever average is around. Okay. Thank you very much, Doctor.
Hello, sir. Good evening. Very good webinar by you. And thank you for organizing. I am Dr. Sai Ganesh, professor in the periodontology department. I want to ask one question, sir. What you told, biomodulation and biostimulation both are same, sir, because we are also using a low-level diode laser for the healing of after doing gingivectomy, after doing the flap or the other things. We are also doing the research in this thing. Whether both the things are same, sir, or biomodulation is little bit different from this thing, sir. I want to have a clear idea about this thing. Please, sir, can you enlighten me and guide me, sir?
Yeah,
I am new to this.
Absolutely. So that's a great question. So when we talk about photostimulation, we're only talking about trying to promote healing or promoting tissue regeneration. So that is only one part of the coin, right? So that by using a particular dose, you can stimulate healing or stimulate regeneration. And periodontists, as you're very familiar, are very good at doing that after they do scaling and root clearing. But in the case of mucositis, there is actually pain and inflammation. So if you use the same device in a different protocol, you can also inhibit all the pain and inflammation. And that is why we like to use the term modulation, because if you use the right dose, you can inhibit certain processes. And if you use a different dose and a different protocol, you can stimulate certain processes. So
very good answer,
just
sir.
the terminology.
Little bit more enlightenment, sir. Can we use this in the perio also, sir?
Yes, absolutely. So one of the major areas of application of photobiomodulation, and we do, again, we are limited by time. So I didn't show you all the data that we have for periodontal disease and implant disease. We have been able to use it very effectively to promote osseointegration, but we have also been able to use it to disinfect and improve attachment levels. So if you're interested, I'm happy to send you our periodontal research presentation. There are various applications, sir, and depending on how you use it, you can actually use it for various different processes.
Oh, it's very nice, sir. Thank you very much, sir. You have enlightened me very nicely, sir. Thank you, sir. Thank you so much, sir.
Any other questions by any participants? Praveen, I have one question. Is there any clinical trials have been started, or like because or what is the evidence of clinical trials?
Right. So there are several ongoing clinical trials. My lab does clinical trials in medical as well as dental. So just to give you an example, we have an ongoing clinical study on concussion, on traumatic brain injury. But we also have another study on peri-implantitis, as Dr. Ganesh was asking. We are actually doing a lot of studies on periodontal and peri-implant healing and osseointegration. So there are lots of exciting studies all around the world, but at Buffalo, we have a group of clinicians who are very interested in lasers in dentistry, which is surgical lasers, but they're also very interested in non-surgical lasers and photobiomodulation. And Buffalo is the actual birthplace of photodynamic therapy, which is the other type of treatment where you're trying to get rid of biofilms and disinfection. So there is a lot of rationale to combine PDT and PBM in various kinds of diseases, including periodontal disease. There's a lot of interesting work on it.
Okay, that's great.
Dr. Praveen, there is a question in the chat box that is photomodulation effective in the facial skin post-operative scares?
Yeah, so that's a great question. So we have several...
By Dr. Deena Rajguru.
Yeah, so there is a lot of evidence for pre-treatment. So mucositis is a good example. So we don't do photobiomodulation. We can do photobiomodulation if you have mucositis, which is the treatment protocol. But the most effective protocol for photobiomodulation right now is preventive. So you do it even before they get radiation and chemo or transplant. And we have found that it improves the resilience of the patient. There's a lot of interest, especially with the COVID pandemic ongoing, is can we make patients, we can't stop them from being disinfected, right? Because there's no way to keep everybody indoor every time. So can we make people more resilient? And I think the vaccines is obviously one big way of doing it. But we are very interested in, we're actually doing some studies where we are trying to understand if we can boost the immune response and your body's ability to fight infections. And this is true even for surgery, right? There is some fascinating studies with cardiac surgery, people who undergo bypasses and different kinds of cardiac surgery. People have found that they can, if they treat them with photobiomodulation, the amount of stress that is induced in the tissues is lower and the patients recover faster. people have done fascinating studies. This is studies done by Dr. Yuri Oron, and you can look up the literature. I'm happy to send anyone who's interested. He has published several papers on human and animal studies where before doing any kind of surgery, he treats them with light, and he finds that the recovery from surgery is much, much more effective. So it's a whole large field, and you'll be amazed at the kind of research that's going on.
Is there any experiments on dentine regeneration?
Yeah, so that is the central part of my research. So I didn't have time to go into that. So my major research interest is dentine regeneration. And you're all dentists, so we all understand calcium hydroxide pulp capping, or I think people now use MT and EMD very frequently. So all of those pulp capping techniques uses controlled osteodentine formation, right? And we have been able to show that light, and this was a central part of my PhD thesis, is can we use light to do that? And we are actually currently in the process of trying to do clinical trials in that space. And we have found that light treatment in a controlled manner is capable of stimulating odontoblasts to form dentine. And that is definitely something that is possible. And there's lots of good evidences for that.
Okay. Any other questions?
I just wanted to emphasize the good news is photobiomodulation can do a lot of different things. The bad news is it can do a lot of different things. So unless you know how to use your tool, just like every other tool that you have learned, whether it's a new bar or a curette, you have learned how to use it well, right? And that's where photobiomodulation is. And I congratulate your team, Dr. Managutti, because I think having these webinars gets that information out there. So.
Yeah.
Yes, sir, you're correct. And now, as we are having no more questions now, I would request Dr. Anil Managutti, sir, to please present the vote of thanks after this wonderful webinar by Dr. Praveen. Sir, please.
Yeah, thank you, Ankita. It's my, yes, sir.
Before,
sir, Dean, sir.
Ankita, let me appreciate about today's deliberation. Excellent and very informative like deliberation was there by Dr. Praveen. Once again, thank you, Dr. Praveen, for sparing your valuable time for our delegates. And I think you have opened many directions for research also out of your deliberation. So there are many take-home points for our delegates. Thank you once again on behalf of SANKALCHAND PATEL University. Okay, Anil, go ahead.
Yes. So it's my proud privilege to, give word of thanks for this webinar series. So the SANKALCHAND Patel University in North Gujarat, one of its top 20 university in Gujarat, which is providing a medical, dental, all technological education in North Gujarat area. And I would like to thank Praveen R Arany for accepting our invitation and enlightening our participants regarding the photobiomodulation, because this is very new and he is worked for 2 decades in the same field. So you are the best person to enlighten all our participants in this. So thank you, Praveen, for sparing your valuable time, and thank you very much. And I would like to thank our president, sir, Dr. Prakash Patel, sir, who is not there, who encourages all the activities for the national or international level and supports us, and all the guidance and everything is given by Provost sir, V.K Srivatsva sir. So these webinars are not possible without the support of Dr. J.R Patel sir. For any webinars or any workshops, anything you tell, sir is there always day and night to help us. So I'm very obliged, and big thanks to J.R Patel sir for encouraging us and helping us for organizing this webinar. And I would like to thank Dr. Ankita and Dr. Hitesh Rawal and all my department staff, Dr. Shailesh, Dr. Rushit, Dr. Nirav, and Dr. Disha for helping me for organizing this, and PGs also. And I would like to thank all the participants for registering and participating in this webinar. And if you have any queries or anything, you can just email us, or you have the email, and you can send us on the text also. We will try to reply for that. Thank you very much. Thank you.
Thank you, everyone.
Thank you. Everybody.